today we'll discuss a brief paper on these two genes, Rin3 and BIN1. BIN1 we've seen already as a molecule that facilitated calcium influx into neurons and therefore indirectly facilitated the phosphorylation of APP and tau, both of which contribute positively to the formation of both plaques and neurofibrillary tangles. But today we'll talk about not a completely unrelated function of BIN1 and its relationship to this other gene called Rin3. So here's the paper that we'll discuss today. The neuronal specific isoform of BIN1 regulates beta secretase cleavage of APP and A beta generation in a Rin3 dependent manner. It was published in Scientific Reports. Scientific Reports is one of those journals that will publish very brief papers that are of high significance. So you don't have to have a lot of data and a lot of findings to publish in Scientific Reports, but the bar for novelty and importance is pretty high. And this paper meets that bar because, as the authors explain in the introduction, why is this? As the authors explain in the introduction, I'm going to skip the F first and then I'll come back to it. BIN1 is a gene that has generated a lot of interest in the context of Alzheimer's disease research because it is the second gene with the highest correlation to susceptibility to developing Alzheimer's after ApoE. So when it comes to sporadic Alzheimer's disease, that form of Alzheimer's disease that is not familial, that is not related to mutations in genes, BIN1 is the second to ApoE to show a significant association with developing Alzheimer's. So there's been a lot of interest on what BIN1 does. So let me take a step back now and mention that in the intro, the authors explain how APP is processed by base 1. Base 1 is really another name for the beta secretase, one of those original secretases that will cleave APP first to generate an intermediate that then gets further processed by gamma secretase into A beta peptides. So base 1 will cleave APP mainly inside of RAP5 early endosomes, RAP5 positive early endosomes. The RAP5 detail, it'll come up later, but don't worry too much about that now. It's just a type of endosome where beta secretase will find APP and cleave it. And the authors emphasize that endocytosis of APP is crucial for A beta production. If APP is not endocytosed into these RAP5 endosomes, then it won't be cleaved to form A beta peptides. So as we just said, BIN1 is important from an epidemiological point of view. No one really knows what BIN1 does. And this paper will explore an aspect of BIN1 that is unknown as of yet, and that is that BIN1 has many different spliceoforms. So if you guys remember back from our conversations about alternative splicing, the BIN1 gene is transcribed into one pre-mRNA, but then that pre-mRNA is alternatively spliced into different isoforms. In this case, there's 10 different variants of spliced BIN1 mRNA, V1 through V10. And those variants, in this particular case, will generate different proteins, right? We discussed when we talked about alternative splicing that not always will you get different amino acid sequences in the final protein, that sometimes alternative splicing will affect other aspects of gene expression. But in this case, these variants of BIN1 do contain different domain structures, right? So in this case, what's of most interest to these authors are V1 that is shown here in a cartoon manner. So there is these domains, and notably the big difference between V1 and this other version that they're interested in, V9, is that V1 has a clap domain and V9 doesn't. Okay? Why do we care about V1 and V9? Because research has shown that V1 is expressed in neurons and astrocytes, whereas V9 is expressed in oligodendrocytes and microglia. And once again, this recurring theme that whenever you see specific patterns of expression, you can start hypothesizing about specific roles of these proteins in various cells. The thinking behind this, once again, is that if there was no specific function for expression of a given variant in a cell type, then you would have expected that throughout evolution, that specificity in expression would have been selected against, right? So whenever you species go through the trouble of putting in place a whole bunch of mechanisms for specifically expressing a protein or a gene in a given cell type or under a given set of conditions, the thinking is that there must be a reason for that provided that selection pressure to make sure that those mechanisms, which tend to be costly and complicated or complex, stay in place. Does that make sense? Right? So anytime that we see patterns of specificity in expression, there is usually the hypothesis accompanying those observations that there may be a reason why those isoforms are expressed in the way they are. Questions so far? Okay. So, not only is the expression of V1 and V9 cell type specific, with V1 in neurons and astrocytes, V9 in microglia and oligodendrocytes, but also research has shown that during AD progression, there is, when you take a look at the total amount of BIN1 gene being expressed, there is an overall increase in the expression of BIN1. But when you start looking a little more closely into the relative amounts of the different isoforms throughout AD progression, what studies have shown is that the expression of V1 decreases, whereas the expression of V9 increases significantly. So overall, yes, the overall amounts of BIN1 are going up, but the amounts of BIN1 V1, that V1 variant, are going down. And that led to a hypothesis based on data, obviously, on some observations, that V1 acts as a negative regulator of RAP5-mediated endocytosis and tau pathology. So what that means is that V1 seems to somehow inhibit RAP5 endocytosis. And if you recall from the previous slide, RAP5 endocytosis of APP is critical for allowing for APP processing and the generation of A beta peptides. So these new hypotheses came up linking the presence of V1 to the processing of APP and A beta. So before we go through the hypothesis, let me tell you real quick that there is this protein called Rin3, the other gene that is of interest to the authors in this paper, that is GEF or GEF, and that stands, we've seen this before, but that stands for GDP-GTP exchange factor. So there's this very large family of proteins called GTPases or G proteins, and RAP5 is one of them. So RAP5, being a G protein, can exist in these two conformations. It can exist bound to GDP in which case RAP5 will be inactive, it will be off, it won't really be doing much. Or it can exist as a RAP5 bound to GTP. Okay? And it is in that state that RAP5 will do things. In this case, it will mediate endocytosis. Now, G proteins have the ability to hydrolyze the GTP they're bound to, so they can go back to their inactive state by themselves. Sometimes with a little help, but for the most part, they can hydrolyze the GTP they're bound to, and then they go back to their inactive state. So for them to be turned on again, they need help exchanging that GDP for GTP so that they can become active again. And that's where the gaps come in. That's where the GDP-GTP exchange factors come in, like Rin3. So Rin3 is helping RAP5 swap a GDP for a GTP and be turned on. There is where BIN1-V1 is thought to come in by sequestering Rin3, sequestering it away from RAP5, right? Such that Rin3 couldn't do that swapping of GDP for GTP in RAP5. And that would, if that is the case, that would keep RAP5 inactive. Okay? So let's put everything together. Observations have shown that during AD progression, there is a significant decrease in V1. If V1 is sequestering Rin3 and keeping RAP5 inactive, then the decrease in V1 expression would free Rin3, which could go then find RAP5, do the swapping of GDP for GTP, activate it. RAP5 could now do endocytosis and facilitate APP processing. So the thinking is that by downregulating V1, now you're helping RAP5 do endocytosis of APP, creating more A-beta peptides and obviously worsening progression of Alzheimer's disease. So all of this sounds great, but the truth is that nothing is known about what BIN1-V1, the V1 allele, or V1 isoform, I should say, and A-beta metabolism. And that's what this paper is addressing. Right? So it's a very important question that is outstanding in the field. We know that BIN1 is really important for epidemiology of AD progression, particularly the sporadic form. And we know a few things about what BIN1 could be doing in cells, but this connection to APP metabolism and A-beta processing was not known, so the paper addresses this, and even though it's just a few figures, it's strong enough and impactful enough to make it to scientific reports. Okay? Questions, comments? Good. Okay. So let's get into it. The first experiment they do is to look at where V1 and V9 are located in cells when there is a lot of Rin3 or not expressed in those cells as well. So in this experiment, what they did is they took cells and they overexpressed either a GFP tagged version of V1 or a GFP tagged version of V9. Okay? So when they're looking at GFP in these cells, what they're really looking at is where V1 or V9 are, respectively. In these cells, they also expressed a RAP5 that is tagged with another kind of fluorescent protein called M-cherry. And it's called cherry because this thing will fluoresce red. So in this case, whenever they're looking at M-cherry fluorescence, what they're looking at is RAP5, where RAP5 is. And finally, they overexpressed in these cells a Rin3 protein that was tagged with, it sounds like a joke, but it was tagged with tag, or actually flag. Flag is one of the common tags that researchers can associate to proteins. So there's flag, MYC, HIS, V5, cherry, GFP, all of these things that you can associate to proteins so that you can follow them around. Okay? So with a flag tag, they can follow, and using an anti-flag antibody, they can follow where Rin3 is. Okay? And then the last panel, the merge of those types of fluorescence, simply indicate where all three are together. Okay? Questions so far? Pretty straightforward. They're putting into cells these proteins with specific fluorescent tags, and then basically they ask, where are they? And what they see, and you can probably appreciate, when they co-express V1 along with RAP5 and Rin3, they all end up in the same places, which are presumably, obviously, these early endosomes. So they're all combining into these very localized regions of cells. But when they express V9, everything seems to be scattered around. It's much more diffuse. They say it's cytoplasmic. All of these things tend to be membrane bound one way or another, so it may simply reflect a diffuse staining on the cell membrane rather than necessarily in the cytoplasm. Okay. So I think it's pretty easy to appreciate that from the images, but then they quantified. Yes?
Same location.
So colocalization means just in the same location. Obviously, that would be the first step towards them working together, right? So normally these things are shown such that if you want to make the claim that they're working together, but you don't see them in the same place, it's a tougher sell, right? Whereas if on the one hand you show that they're all in the same place, and it's just not anywhere, they actually seem to be really in the same place, then it's much easier to hypothesize that they're actually also working together. Does that make sense? Yeah? Any other questions? All right, so the bar graphs to the right are simply quantifications of what the microscopy images showed. These are confocal microscopy images. And yeah, as we can appreciate with our own eyes, what they see is that V1 is much more frequently co-localized with Rin3 and with RAP5 than V9. Right? V9, it's only somewhere around 5 and, well, maybe a good 20% or so of the times that V9 co-localizes with these things, but V1, the rate of co-localization is significantly higher. Okay? I copied the legend to the figure showing this data from the paper directly because it's pretty explicit and self-explanatory. It says the neuronal isoform V1, but not V9, translocates to RAP5 positive early endosomes in a Rin3 dependent manner. There are some data in the same figure that I didn't show you. So these are panels C and D or something like that. There are some previous panels in the same figure showing that if you do not overexpress Rin3, none of this happens. Okay? And hence the Rin3 dependent manner. Now, one question that I wanted to discuss with you, I'm going to give you a few minutes. So this is not going to be one of those that I ask a question, I expect an answer right away. I want you guys to sit with this data and address this question now here. So the authors conclude that Rin3 is required for V1 to be recruited into RAP5 endosomes. But what else can we say when you look at this data? It's an open-ended question, and it's okay if you want to consult with your neighbors. But we look at the data and they conclude something that is supported by the data, that V1 is being recruited to where RAP5 is in a Rin3 dependent manner. But what else can we say? The comment here, the answer is Rin3 is recruiting V1, but not V9. So maybe Rin3 can interact with V1 because of some domain that V1 has and maybe V9 doesn't. Is that it? Okay. That's so correct that we, in fact, we'll use a couple of slides right after this to address that point. But it doesn't come from this data, right? These data are simply showing where these things are in cells, and the authors concluded that Rin3 brings V1 to Rab5 endosomes, but it cannot do that with V9. What I was asking you to do is maybe go a little further by looking at the data and see if we could say some additional things, all right? What you brought up is a mechanistic explanation of what they have already concluded. It's taking the research a step further and saying, now that I've concluded that V1 is being recruited, but not V9, why might that be, right? What I'm inviting you to do is ask yourselves, what else could we say based on this data? Because in my opinion, the conclusion is not incorrect, but it's slightly incomplete.
Just looking at the graphs, when the Rab5 is present, it seems that there's an increase in the amount of V9 that's colocalized.
Mm-hmm.
So maybe that's a point. It's not necessarily that it's entirely not colocalized, but less efficient.
Okay, good. So V9 is recruited, but less efficiently. Oops. Efficiently. See, there's looking a little deeper into the data here because we're going into the graphs and saying, hey, it's not like there's no recruitment at all, it's just less efficient. Jacob?
The activation of RAB is not like entirely dependent on V1.
Say it again, the activation of RAB.
Yeah, like it'll still show up even if there isn't any, well not any, but if there isn't that many V1, V2.
And when you say activation of RAB, what do you mean?
It's just like the mechanism of it.
I'm having a hard time hearing because
Sorry,
of...
I'm having a couple of
I like where you're going. So what you're saying is there's something about RAB, right? Rab5. And then you said that it's not entirely dependent on V1. Is that what you said?
Yeah.
Yeah? And where do you see that? Or where? So when V1 is present, where is Rab5? Hmm? Yeah, they're always in the cell. So a little more precise. They're always in the cell, but when V1 is present, where is Rab5? I'm really sorry, but I think there's a slight confusion here. V9 is BIN1, and V1 is BIN1. There are two flavors of BIN1. There you go. So we go back to this idea that the reason why V1 and V9 may be recruited to Rab5 endosomes or not, maybe you mentioned the presence or absence of certain domains. Now you're taking a step further, saying the shape of them. I agree, but that refers more to mechanisms explaining the observation than additional conclusions or additional observations that we can make.
Well, I guess they didn't show BIN1 or BIN1.
Finally, yes. So I'm going to take it back to what you said at the very beginning, that V1, you said something like V1 is required.
Yeah, that's what authors like conclude, but that's still not.
What the authors are concluding is that Rin3 is required to bring V1 to the endosomes. But what else are these very same data showing us? That when you have V9, Rin3, which we can follow with the anti-flag staining, and Rab5, which we can follow with the M-cherry, are all scattered themselves, right? So it kind of goes both ways. The authors say Rin3 is required to bring V1 to Rab5 endosomes. Okay? But I could turn that around and say when you don't have V1, Rab5 and Rin3 are not together anymore either. If we zoom in on this thing, whenever two things are together, or in this case, three things are together, you'll see white pixels, right? Because you have some green fluorescence, some blue fluorescence, some red fluorescence. You combine them all, and that makes near white light, right? And you can totally see that here. In all of these spots where all three combine, you see these quote-unquote light, white light coming out. It's not really white light, it's probably some sort of far yellow, but the point is that they're all combining. The computer or the microscope can detect all three fluorescences, and it says in all of these pixels, I saw the three colors. But when we go to this, now you see a lot of blue colors, a lot of red colors, and a lot of green colors that are by themselves. And that's an indication that they're not in the same places. What's interesting, I think, is that the authors made a big deal about how Rin3 brings V1 to where Rab5 was, but they're not emphasizing the fact that without V1, Rin3 and Rab5 are not interacting with each other either. Does that make sense? Yeah? The reason I bring this up is because I think there is room with their data to make the case for a model in which it's not so much that Rab5 and Rin3 were together and then they bring along V1, but instead that when V1 and Rin3 interact with one another, now they go and meet with Rab5. So you have all three, they'll end up together. You don't have Rin3, they won't end up together. That's the image that I didn't show you. I should have probably. I didn't show you. But when Osasi asked, I mentioned that looked a lot like this. And then when you don't have V1, because you have V9, they're not together either. So it's not like Rin3 and Rab5 are already buddy-buddies and they bring V1 into the endosomes. It's more like when the three are together or expressed in the same cells, then they end up together. But if either V1 or Rin3 is missing, they won't. Okay? Cool. So, now let's talk about those mechanisms, right? So, they concluded that Rin3 brings V1 to Rab5 endosomes. And as you were suggesting before, it may have something to do with domain structure. V1 has a clap domain, V9 doesn't. So how could Rin3 be bringing V1 but not V9? Exactly. So they look at the domain structure and they think, well, maybe Rin3 is binding to V1 through the clap domain. The clap domain is not there in V9. So the only reason that Rin3 is bringing V1 into these endosomes is because it can bind to it, right? So they decided to test that idea, and they did a co-immunoprecipitation experiment. The idea behind any co-immunoprecipitation or co-IP experiment, and if you've heard this before, I apologize, but so that we're all on the same page. Whenever you're probing the interaction between proteins in a given biological sample, if you have an antibody that allows you to bind and separate a given protein from the mix, then you can ask what other proteins are coming along with it, right? Now, why do we call it immunoprecipitation? Because what we do is we let the antibody bind its target, and then we precipitate the complex, right, using biochemical tricks. And then what we look for is partners that are co-immunoprecipitating with your target protein. Okay? So that's what they did. And in this case, the IP was using an antibody against GFP. If you guys recall, the BIN1 variants, either V1 or V9, were tagged with GFP. So they have a little GFP attached to them. So you can use an anti-GFP antibody to bind that GFP and bring the whole thing down. And then the B here stands for blot. So what you do is you bring down GFP, therefore V1 or V9, depending on what was being expressed in the cells, and then you probe for the presence of Rin3. Okay? Let's walk through this because this is a very typical schematic or very typical presentation of co-IP data. So in the bottom plot, what they're showing us, and I believe this may have been a bit of a typo, so they probably meant IP, anti-GFP, blot, anti-BIN1. So what they're doing is they're bringing GFP down, and then they're probing for BIN1. Mind you, GFP is genetically fused to BIN1 in these constructs, right? They've introduced these fusion proteins into the cells, so it's no wonder that when you bring down GFP, you will bring down along with it BIN1, right? Either the V1 variant or the V9 variant, depending on which sample you're looking at. And that's what we see down here. They bring down GFP, they look for BIN1, and sure enough, they see V1 or V9, V1 or V9. The difference between the left side of the panel and the right side of the panel is the right side of the panel, they added Rin3, right? And when they add Rin3 and they bring GFP down, now they see that Rin3 is coming down with both V1 and with V9. And the amount of Rin3 that is coming down with these BIN1 variants seems to be the same. When they did densitometry and quantified, it was the same amount of Rin3 that was coming down. So these original hypotheses that maybe Rin3 is bringing V1 to endosomes because it can bind to it, perhaps through the CLAP domain, but it cannot bind to V9 because V9 doesn't have the CLAP domain, kind of goes out the window, right? Because what they see experimentally is that when you bring down BIN1, either V1 or V9, you bring down the same amount of Rin3. They do have an explanation for that. Turns out that separate research has shown that the most likely place for Rin3 BIN1 interaction is through the SH3 domain of BIN1, which happens to be present in both V1 and V9. So that would explain why when you bring down BIN1, you will bring down the same amount of Rin3, whether you're working with V9 or V1, because both of them have the SH3, both of them will have the same capacity to interact with Rin3. And then they have a model that they present in the discussion, but that I illustrated here. Their model is that the SH3 domain can bind to the CLAP domain in what they call an intramolecular interaction, meaning an interaction between two domains of the same molecule, of the same BIN1 protein. What they're proposing is that when there is no Rin3, that intramolecular interaction between SH3 and CLAP takes place, and that sequesters CLAP away from interacting with anything else within the cells. But when they add a whole bunch of Rin3, which they did here, now Rin3 binds to the SH3, steals it away from CLAP, and what happens now? CLAP is free to go bind to whatever else it can. And what they're proposing is that these interactions between CLAP and other things is ultimately what's bringing BIN1 to the endosomes. Okay? Now, if you put a lot of Rin3 in cells that express V9, the interaction between Rin3 and the SH3 in V9 will still take place. But there is no CLAP to be released or freed by that. So V9, its SH3 may be bound to Rin3, but that doesn't make CLAP all of a sudden appear in the V9 structure. So V9 does not have CLAP, and therefore it will not be brought to the endosomes.
So shouldn't you see on the fluorescence on the previous page,
Mhm.
Rin3 and BIN1 interacting then, if it's still able to bind?
Let's go back because what you're saying is, if that is true, what would you see then? Walk me through it. Say it again, because
Green plus blue.
green plus blue, right? So you expect to see a lot more green plus blue, which would show us probably as a cyan, fluorescence-wise. Maybe if we squint our eyes. Now, but let's go to the quantifications. They did quantify V9 and Rin3, and there is very little. So you're absolutely right, they should have. Hold that thought, because this reinforces an idea that I had coming into today's lecture. I hadn't thought of that, and you're absolutely right. The model would be beautiful if these both of these bars are high, but then only this one is short. I like that. Abby?
It's not any of the other domains that they share.
Because so how do we know that it's not any of the other domains that do what?
That bind to CLAP. Like here we have 4.
Oh, excellent. That's hypothetical. It's based on the fact that other research has shown interaction between Rin3 and SH3 domains. Yeah, and the references in the paper, I don't know if it was the SH3 domain of BIN1 or if it was an SH3 domain of some other protein. The thing about these domains, SH3 domains, CLAP domains, SH2 domains, blah, 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 all of these domains happen in different proteins throughout the genome in various combinations. So we've seen SH3 domains before in the context of B1, but trust me, BIN1 is not the only protein that has SH3 domains. So it could very well be that previous research has shown interactions between Rin3 and SH3 domains, and that leads them to, among the various domains that BIN1, both V1 and V9 have, SH3 domain is sort of the higher or the most suspect, I suppose. No? Good. We'll have to think about that. Yeah, that's a good one. Cool. So, now they're working with this model that Rin3 is simply freeing CLAP, and CLAP will, through binding to who knows what, facilitate the recruitment of V1 to the RAP5 endosomes. So remember at the beginning of the paper, they say that we know that BIN1 must be important because it shows up right after ApoE in epidemiological studies, so it must be doing something. And we know a little about what BIN1 may be doing through regulation of endocytosis to tau pathology, but that we don't know about what it might be doing to APP processing and A beta pathology. So they decide to address that next. And in this experiment, what they're doing is they're simply measuring by ELISA how much soluble APP is being produced by cells that express, and I'm going to walk you slowly through it, that express nothing. That's what we say when the authors say GFP, what they're saying is GFP that is not fused to anything. V1 or V9, that I should clarify, are fused to GFP. Right? Or V1 and V9, fused to GFP, co-expressed with Rin3. Okay? So the way to read these plots is pretty straightforward. The bottom panel, we have the total APP. And that's just a measure of when you're working with these cells and these samples, there's always going to be some cells that shed off some pieces of membrane. They may, the cell as a whole may break up because it died and it's releasing some of the fragments of membranes with the various things that they had when they were alive. So all of that can happen, and it serves as some sort of baseline or background. And when they measure the total amount of APP that is present in the conditioned medium, that's what CM stands for, in the medium floating around the cells, they see that the levels of total APP are the same. So that gives you an idea that these cells are expressing similar amounts of total APP. But when they look specifically for amounts of APP beta, meaning a fragment of APP that has been cleaved by base 1, the beta secretase, what they see is that there is essentially only one condition in which there is a significant reduction in the amount of soluble APP beta, and that is when V1 is co-expressed with Rin3. If you're wondering, well, what about that other condition here? There's another lane there with a lot less of this soluble APP beta. That's when they treated the cells with a base 1 inhibitor. Okay? So when they purposely inhibited base 1 activity, they would have expected a drastic reduction in soluble APP beta, and that's what they saw. But then the only other condition in which they saw drastic reduction in base 1 cleavage of APP is when V1 and Rin3 were expressed together. Okay? This goes really well with the idea that V1 inhibits early processing of APP and might therefore inhibit later processing of APP beta into the toxic form. Right? When APP beta gets cleaved by gamma secretase, it will generate the toxic A beta 40 and A beta 42. And that's what they measure in these graphs here on the right. They measure the amount of A beta 40 and A beta 42 produced by the cells when they had nothing in them, when they had V1 alone, or when they have both V1 and Rin3. So when they have both V1 and Rin3, there is a drastic, statistically significant reduction in the amount of toxic A beta products. Okay? Questions or comments, anyone? Yeah.
So that other thing you said is the V9, V1, the last line.
Mm-hmm.
So it's like some kind of control?
Correct. It's a positive control. So essentially, the idea is they're using this technical or this experimental approach to see if there would be reductions in the amount of APP beta produced. When you go into an experiment, you never know if you're going to see changes in what you're hoping to see changes. So what do you normally use? A positive control, something that you know should show an effect. So you use that as a positive control, and in this particular case happens to be very similar to what you see experimentally, right? The reason we introduce these positive controls is, let's imagine for a second that that band was super dark, thick and dark. Then the experimenters may have thought, well, our hypothesis may still be correct, and it's just that our technical approach was not good. So that's when you use a positive control, just to make sure that you are technically able to see what you're expecting to see. And then you also use a negative control, and in this case, the negative control would be any of these samples here, right? So you're basically saying, what should I expect to see if there is no change whatsoever? Okay, so I'm once again copying verbatim the figure legends. Figure 2, where this block comes from, says the neuronal isoform V1, but not V9, reduces beta secretase mediated processing of APP in a Rin3 dependent manner. You can see here clearly the first 3 panels show no effect, but then the second 2 panels do, and that's because that's where we added Rin3 to the neuronal culture. And then figure 4, where these bar graphs come from, co-expression of Rin3 with the neuronal isoform V1 in cells reduces A beta generation. Okay. Cool. So once again, they're like, okay, but what is the mechanism behind this reduced APP processing? So far, I see that V1 goes into endosomes when Rin3 is present, but how does that explain that APP is being cleaved less by base 1? So far, I see V1 going into these things, and I see that less APP is being processed. How do we connect the two? So it turns out that going into this experiment, the authors formulate a hypothesis based on, again, previous knowledge. They say, because we know that APP processing depends on endocytosis into endosomes, perhaps the reduced APP processing is due to reduced internalization of APP. In other words, reduced APP endocytosis into these endosomes. And they're putting everything together. Rin3 brings V1 to endosomes, and less APP is being processed. Maybe the presence of V1 somehow delays or inhibits the endocytosis of APP into these things. So here's the experiment they do. So it's a pretty clever experiment. They take their cells, and first they incubate those cells with reagents that will biotinylate all surface molecules. Biotin is a short peptide that can be biochemically added to other proteins when you have a biotinylating enzyme present. So essentially, you can take your cells and incubate them with biotin and a biotinylating enzyme, and that enzyme will take this biotin and stick into all of the proteins that are present on the cell surface, on the outside. Why? Because these things can't get into the cells to do that. They can only exist outside. So first you biotinylate a whole bunch of proteins on the cell surface. Then you let some time go by, and you allow the cell to do endocytosis, as it normally would, and some of those biotinylated proteins will make it into the endosomes, right? Then you treat the cells, you wash away the biotinylating enzyme and the biotin, and then you purposely treat the cells with a debiotinylating enzyme, an enzyme that will do the opposite, will go and chop off any biotins that were present on the cell surface. So you debiotinylate the surface, but anything that was internalized is now protected. And last but not least, you crack these cells open, pull down the biotinylated proteins using a protein that can bind to biotin and bring these things down. And then you ask the question, what is coming down with those biotin molecules? So The logic behind this experiment is anything that was on the surface was likely biotinylated. Anything that was internalized will still be biotinylated. Then I shave the surface from any biotin, and I bring anything that was biotinylated down, and then I'll ask, what came down? Right? So it's a way to measure internalization of things that were at some point on the surface of the cells. They did that, and they show us two experiments. One in which, after bringing biotin down, they looked at APP. In other words, to see how much APP had been internalized in this experiment. And then a separate one in which they look at base 1 to see how much base 1 had been internalized in this experiment. And what you see here is actually a time course. So they have cells with V1 only or with V1 and Rin3. And in both cases, they look at time 0, 10 minutes after adding the biotin and doing the whole, washing the biotin away and shaving the surface and so forth. So 10 minutes after they started the process, or 30 minutes after the process. And the way to read these plots is when the cells are expressing V1 only, at time 0, there is very little internalized. You just started the experiment. 10 minutes after, you'll see an increase in APP that has been internalized and base 1, right? So these things, you have to give them some time. So after 10 minutes, there's going to be more of both, of APP and base 1, that were inside of the cell and that were protected of all of these biotin shedding. After 30 minutes, there is little of both again. And the interpretation of that is that everything there was has been internalized, so there isn't really anything else to internalize anymore. That is if they expressed V1 only in the cells. When they express V1 and Rin3, and now V1 can go into the endosomes and interact with Rab5, blah, 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 what they observed is that the internalization of APP is delayed. It used to happen 10 minutes after starting the process. Now you can see it, but only after 30 minutes. Base 1 internalization, on the other hand, seems unaffected. Base 1 is being internalized into the cell, regardless of whether or not V1 is being brought to the endosomes or not. And these graphs on the right are simply numerical representations of several repeats of these experiments. And what you see is that when the cells, it was not in the legend, but I'll add it here, when the cells express V1 only, the bulk of APP is internalized after 10 minutes. But when the cells have V1 plus Rin3, the bulk of APP is internalized after 30 minutes. And the curves for base are very similar in both cases, with a slight increase after 10 minutes and then a drop at 30. So what the authors conclude from this is that co-expression of Rin3 with V1 delays cell surface endocytosis of APP. Okay? I think we have enough time for this slide for sure. This is the last figure in the paper where they present their model in a graphical form. Essentially what they say is that when cells do not have Rin3, that's the left panel, there's going to be some endosomes that endocytose base 1. There's going to be some endosomes that endocytose APP. And they'll come together to form these early combined or fused endosomes that bring together base 1 and APP. So base 1 will go to town with APP on APP, and it will start creating APP beta, which will then be cleaved into the toxic A beta forms. That's when there is no Rin3, right? What they're arguing is that when there is Rin3, what's happening is Rin3 will go and interact with the SH3 domain of BIN1, freeing the clap domain in BIN1. That is BIN1 V1, V9 doesn't have it. That clap domain will interact with a molecule called clathrin that is important for the endocytosis of APP. So now what BIN1 V1 is doing is competing with APP for clathrin. It's like, oh, no, this clathrin is mine. So now V1 ends up inside of the endosomes and APP is left out. So when these endosomes form, what will you see together? V1 and base 1. No APP, no cleavage, no soluble APP beta, no A beta 40 or 42. That makes sense? Yeah? So that's the model. I have one more slide that I suppose we can discuss briefly on Wednesday because we're running out of time. But essentially, if all of this is true, then I suppose one could say that V1 inhibits not the catalytic activity of base 1, but the function of base 1, in that it's stealing away its substrate. So V1 is inhibiting base 1 in the sense that it takes its substrate away. And base 1 normally would produce a lot of A beta oligo, but if there is a lot of V1, that's going to be indirectly inhibited. When you inhibit an inducer, you're essentially inhibiting the process, right?